project Biocash, and it's a cooperation between ILVO, Hasselt University, and Sebastis and Spain. So there are a lot of challenges for increasing the sustainability and circularity of soilless cultivation. So these challenges are listed here, and biochar can really help to work with these challenges. So biochar is actually a renewable material. It can also allow to better or more efficiently use nutrients, organic fertilizers. You can also try to adapt the cultivation practices during the first time you use a horticultural substrate in order to allow to reuse or upcycle it for more than one cultivation cycle. So really to have circularity and soilless cultivation. And after all these cultivations, you can still use the biochar for carbon storage in the soil. So as an end-of-life stage of the spent growing media with biochar, it's really also promising to have biochar in it. So biochar may help to increase sustainability of horticultural substrates, but you can use biochar in different applications. So you can use biochar in very low doses, so some grams per liter of growing medium, just for disease suppression or as a carrier for biocontrol organisms, or as a carrier for nutrients, or you can use it as a liming agent in very small amounts. If you want to increase the amounts, you can also use the biochars as a fertilizer. You can further increase the amounts of biochar in the growing medium, and then you can use it, for instance, for perlite replacement. So the white spots you sometimes see in growing media, they are actually clay particles which are heated at very high temperatures. And these perlite particles you can replace perfectly by biochar particles. Or you can further increase the amount of biochar, for instance, to replace bulk materials like peat, coir, or other materials by renewable biochar. But the big dream is, of course, that maybe one day we are able to grow plants on 100% biochar. So we have biochar as a standalone horticultural substrate. But biochar can also have relevance for growing media, because you can use it as an upcycling strategy. You can convert spent growing media, which are like the waste materials of soilless cultivation. You can use it to upcycle it into biochar. Or you can use this biochar production to get rid of pollutants, pathogens, at least reduce their presence, or even completely get rid of them, also including microplastics. But it can also be part of a holistic system where you use the pyrolysis for heating greenhouses, for CO2, for greenhouses, and then the biochar you can use in growing media. So here you have some examples of biochar. So on the right side, you see like a fiber-based biochars, one based on miscanthus on top, and at the bottom you see one based on flax chives. And on the left side, you see biochar based as a recycling strategy. So there we have converted spent growing media into biochar. So one based on peat and one based on coir. So we have different strategies to include the biochar, and you can also see that we have different shapes and particle sizes of biochars. But the big question is, of course, can we use all biochars as a peat replacer in growing media? And for answering that question, we can use a simple scoring system to assess the suitability for using biochars or other materials in growing media, and to assess their peat, fertilizer, and lime replacement value. And this scoring system, it's based on 8 very important characteristics of growing media. We score them between 0 and 2. So 2 is the better one, of course. We make the sum of this score, and then you get a result between 1 and 16. And the higher the score is, the better your material can be used for peat replacement, or as a fertilizer, or as a lime replacement in growing media. So for different wood-based biochars, we have used this scoring system, and we found quite high scores. Even one score was for one biochar, we had even a 16, which is very high, of course. Then for fiber-based biochars produced from miscanthus, from flax chives, or from reed, we also had quite high scores. And also for biochars based on spent peat, so after one cultivation, we also saw that in some cases the score was very high, above 10. So that was promising, is promising. But for peat-reduced growing media, we saw that the biochar had a lower quality for peat replacement, which is a pity, of course. And then for nutrient-rich materials, we saw also lower scores in general. Even if we produce biochar based on digested or manure, we had very low scores. So it indicates that not all biochar can be used for peat replacement or as a standalone material. Some of the biochars you can really use as a fertilizer maybe, or as a liming agent, but then only in very small amounts. But then when we look at the promising biochars, we see that mainly the pH and the inorganic carbon content of these biochars is still a bottleneck. So these are the issues that we need to solve. And that's what we are going to do today. So it's very important that we know that biochar contains a lot of carbon. We all know, of course, the organic carbon, but biochars may also contain sometimes up to 2% of inorganic carbon on dry matter basis. And we have seen in previous research that the acid buffering capacity of biochars, it's very strongly positive correlated with the inorganic carbon content of biochar. So a biochar with more inorganic carbon, it can really, it has a very high acid buffering capacity. And just to indicate what it might mean, if we have a wood-based biochar with 0.5 grams inorganic carbon per liter of biochar, it's the same actually as like 4 grams of calcium carbonate per liter biochar. And if you then know that we sometimes add only like 2 grams of lime per liter growing medium, it really indicates that some materials, they really can replace lime, or they can even increase the pH of growing media beyond the optimal range. So too high pHs actually. And a very simple example to show that when we mix like 20% of biochar with unlimed peat with pH 4, and then we measure the pH after 10 or 30 days, we saw for this wood-based biochar with the inorganic carbon content that the pH increased by 0.4 after 10 days already, and then with 0.7 after 30 days. So really it indicates that this inorganic carbon, it's an important characteristics of biochar for use in growing media. It can be positive because you can maybe replace lime, but it can become negative when the inorganic carbon content is too high. So here you see some biochars that we have picked from spent growing media, where we have first included, of course, the biochars. And we wanted actually to see what happens with biochar when you use it in a growing medium. So we had 3 full-scale trials where we had 10 volume % of biochar included. And then we compared the properties of the biochar after the cultivation with the initial properties of the biochars. And it was very nice to see that the pH of the biochar decreased from 9 to 6 on average by using it in growing media. And also the nitrogen content increased, and also the mineral nitrogen content was clearly higher. And even there was a small increase in the cation exchange capacity. So it indicates when you use biochar in a growing medium, the quality of the biochar for use as growing medium is increasing. So that's very nice. But of course, when we want to start to grow plants on pure biochar, yeah, it will initially have a pH of 9, and that's not a solution at this stage. So we have to look for other solutions. So we are in a transition from peat-based horticultural substrates to renewable horticultural substrates. And in these peat-based ones, we typically need lime to increase the pH to the optimal range for plant growth. While actually, when we go to renewable materials, we mostly see a risk for too high pHs, and we need like acidification of the materials rather than liming. And one very easy method is using elemental sulfur. And we have tried it for different applications. So we used between 0.5 and 3 grams of sulfur per liter material. And then we saw mostly a pH decrease within 4 weeks. So we did it for substrate blends, which had too high pH before cultivation. We could decrease the pH of these blends. But we also used it for spent growing media in order to reuse them. And we saw that pH was sometimes too high. We could decrease it by using a small amount of sulfur. And we even tried it for composts to decrease the pH before blending them in growing media, or for processed grass fibers, which had a too high pH, we also were able to reduce the pH with a small amount of sulfur. And now the question of today is, can we also use this technique to reduce the pH of biochar? Okay, but maybe first I will explain what is the biological acidification with elemental sulfur. So it's actually a biological process. You can say it's a living acidification because you need bacteria to mineralize the elemental sulfur into sulfates. But of course, these sulfates, they then have a chemical effect. So this sulfur is converted to sulfates in an aerobic environment. So we need oxygen, of course, and we need water. And in these conditions, we have the sulfuric acid, which is formed, and it attacks the calcium carbonates, and these are converted into calcium sulfates, water, and CO2. So it's actually a biological process with a chemical effect. And yeah, we see that based on this equation, we can see that we need lower doses of sulfur when we have lower inorganic carbon contents or a lower carbonate buffer in the materials. So here you see a picture of the elemental sulfur. So it's really a very fine yellow powder, so that you can easily mix with materials in a very small amount. So it would maybe be promising to also have this technique available for biochars, and then it would allow us to use 100% biochar as a standalone horticultural substrate. Because then, yeah, biochar is a very stable material. It's very promising for one cultivation, but it also allows to maybe reuse the substrate multiple times without big problems on the substrate quality. But for doing that, of course, we first need to reduce the pH of the biochar, and we should also select specific biochars. So maybe mainly wood-based biochars or fiber-based biochars with low nutrient contents, a low acid buffering capacity, and the target of this acidification would be to reach a pH of 6 or maybe lower. So it is an aerobic process, and it will, of course, result in more sulfates and also a higher electrical conductivity in your material. And that's what we did. So here you see 10 biochars that were acidified with 2.5 grams sulfur per liter biochar during 1 year. And the target that we set was around pH 6. And then you will see that it's a quite slow process. So we see a pH decline in all biochars, sometimes slow. And only for 2 or 3 biochars, we really reached the target pH of 6 after 1 year. So yeah, it's maybe nice, but it's not feasible to do it in practice. We don't have 1 year to wait to acidify biochars. So we wanted to have a faster method to reduce the pH of biochars. And therefore, we tried combinations of sulfur with fertilizers or with sugar. So we had the one treatment was with only adding mineral fertilizer. So it's a multi-mix potting soil fertilizer, which is regularly used in growing media. We also combined these mineral fertilizers with a small amount of sugar. And then we also used sulfur in combination with chitin. So chitin, it's an organic fertilizer, and by applying chitin, you get really a stimulation of microbial activity. And this microbial activity, it results in the release of mineral nitrogen in your substrate. So that's why we call it an organic fertilizer. So the combination of biochar with sulfur was not that promising. So we added like a third spice in our biochar. It was the chitin. So it has a microbial way of working, because you get this decomposition of the chitin into mineral nitrogen. It has a chemical effect because it releases mineral nitrogen. So we did it for 5 biochars. We did these different combinations, and the 5 biochars, they were wood-based, fiber-based, and we saw a clear drop in pH. So as you can see here, for the virgin biochars, we had an pH of 9 on average. And by using these combinations, we saw that we could reduce the pH even below 5. There was, of course, a very strong increase in the electrical conductivity, but which is very important, it's also the time to reach that low pH. So the winning combination was actually the sulfur combined with the chitin, where we saw that after only 8 weeks, we reached a very low pH by this combination. So at that stage, we were still very happy until we decided to remeasure the pH 8 weeks after the first time we measured the pH. And then we actually saw that the pH further decreased to values below 4 and even sometimes below 3. So we got really a very acidic biochar, which is way too acidic to be used as a horticultural substrate. So this needs like some optimization. On the other hand, we also had the biochars with a high buffering capacity, so with the inorganic carbon content. And there we saw that there was no clear drop in pH when applying the sulfur in combination with the mineral or the organic fertilizer. But we saw microbial activity, as we saw an increase in the EC and in sulfate concentration. And also we saw that the inorganic carbon content was decreasing over time. So there was really activity, but it did not yet result in a pH decrease because of the high buffer capacity. And also, in general, this acid buffering capacity, we could see a decrease over time. So it was a slow reaction in terms of pH decrease, but there was really also a microbial activity ongoing. So then we ended up with this one of these biochars. It's the bark-based biochar produced at 400 degrees. And we had a pH of like 3, which is too low. And then we tried to mix it with the original biochar, which with a higher pH. We mixed it in a 1-to-1 ratio. And after 4 weeks, we measured the pH. And we again saw a very low pH. So the acidified biochar, it really had the capacity to further acidify biochars when mixed. So it was still too acidic to be used in growing media. And this acidic mixture, we further diluted it with different types of biochars. So on the left, you see that we further mixed it with 50% of the virgin biochar, so a 1-to-1 dilution. Then we did also 1-to-3 dilution, so only 25% of this acidic biochar was mixed in virgin biochar. It's the same bark biochar. And then the next 2 treatments, it was actually not with the bark biochar, but we mixed it with a biochar with a high acid buffering capacity, so with inorganic carbon in it. One of them was already treated also with some sulfur, and the other one was as it was original. So you see, we mixed it with biochars with a pH between 7.4 and 8.3. And then the acidified biochar, it had a pH of 3. And then after 8 weeks, we measured again the pH. And now you can see that there is some differentiation in some of the blends. So if we diluted only 50%, we had even a pH, again, lower than 3. But when we mixed it, for instance, with the biochar with the high acid buffering capacity, we had a pH of 7. So it indicates that by diluting the very acidic biochar with different types of biochar, we can make a blend with a pH in the optimal range to grow plants on it. So maybe you will ask me if it was maybe too ambitious to apply 2.5 grams of sulfur per liter. So maybe a smaller dose would be better. And that's also what we tried. So we used also a dose of 0.5 grams of sulfur. And for the bark biochar produced at 400 degrees, we see indeed that the pH is decreasing slower. But in the end, I guess we will also end up with a very acidic pH, as we had it also one of 3.6 after 32 weeks. For the other biochar produced at 650 degrees, we see that the pH decrease is slower, and actually it's increasing again after 32 weeks. So maybe that's a promising treatment for this biochar. And then the next step is to do some trials. So we did trials with cuttings, and also cuttings of a plant that really likes a very acidic pH in the growing medium. So it was really a high, well, it was very difficult for testing under these conditions, but it really allows us to see whether what is the most problematic, the pH of the biochar or maybe the high sulfate concentrations. And it indicates that there is potential of the acidified biochar, although there is a high sulfate concentration, but we further need to test it for other crops and other applications. So what you see in the picture, it's actually biochar treated with sulfur and chitin, and then you get some mycelia growing on it. And then the question is it microbial activation, or is it just microbial or biological acidification of this biochar? Well, when we monitored the mineralization activity, we could really see changes in pH, AC, and sulfates because of the sulfur mineralization. So it's really, microbial activity. And also for the chitin, we saw mineral nitrogen release. So it means that there is also microbial activity from chitin, although this nitrogen can be immobilized again by the biochar. And we also saw interaction because the chitin only mineralized when in the presence of the elemental sulfur, and the elemental sulfur decreased the pH faster when chitin was present. So it seems that these two guys, they need each other to be active in biochar. So this brings us to the conclusions. You can say this biological activation, it's a process, it's really low tech. It's a soft treatment. These products we used, they are even allowed in organic agriculture. But of course, you need to make a balance between the pH decrease and the increase in the salinity or the sulfate concentration. It's a microbial process, and it appeared to have a low impact on the surface properties of the biochar. For the biological activation of biochar, so we have seen that elemental sulfur and chitin, they are partners for this microbial activity, but we need to balance the time needed to have the acidification and the final pH we reach. And then, of course, also the EC increase we might have there. Then for the unbuffered biochars, it seems that the pH range of 5 to 6 is like a critical zone, because when we had a final pH above 6, we saw that the pH was increasing again after some time, while when we had a pH below 5, we saw that it was further decreasing to pH even below 3. So it's quite complicated to get the right pH there. So we still need some fine tuning for this application as a horticultural substrate, but maybe this biological activated biochar can also be applied in other applications. So I would really like to thank the people from the BASTA consortium. It was an FWO funded project that finished recently, and also the partners in the EGP soil project Biocas. And I would also like to thank Rianne Vesser from TNO, because she provided in the HortiBlueC project also several very high quality biochars for part of the trials. So thank you for your kind attention.
Thank you very much, Bart. That is a very interesting topic. I was hooked when you said you have this grading system for biochar, so that was very interesting for me to see. The floor is open to questions, so you can either write them into the chat or you unmute yourself.
Hugh McLaughlin has a quick question.
Yeah, sure.
Bart, by the time we make a gigaton of biochar, we'll have a gigaton of wood vinegar to give away. And I'm wondering if you gave any thought to maybe using wood vinegar as a buffering opportunity to neutralize the char to the target pH.
Yeah, I think that there might be other solutions to obtain the optimal pH for the biochars. The promising or maybe the challenging thing was to do it in a really microbial way, and these trials. But yeah, I think there are maybe other treatments that might be promising as well. Yeah, it's a good suggestion.
Maybe I shoot my question next. So I was wondering about the blends. So if you blend a high pH biochar with a low pH biochar, would you expect that the low pH biochar would still have the same low pH effect if you plant something in it? Because a blend is basically you still have the physical products in the soil with the low pH, and the pH measurement is usually a bulk measurement. So I was wondering if there's any thoughts on that.
Yeah, I think it's indeed if around the root zone, if you have another pH than in the bulk material, it can really change a lot of things. But on the other hand, plant roots can also affect the pH. And also when you add fertilizers, it can also change the pH. So yeah, it's where we are now at the beginning of the plant trials. I think still a lot of aspects needs to be covered by future research about this interaction between plant roots, between fertilizers, and maybe other additives. Yeah.
You got a question from Luke. So the 650 degree biochar is different, shows different results from the 400 degrees. Can you just quickly explain again what the reason is for the different results?
Yeah, I think the higher you go in the pyrolysis process, the more inert your carbon and your biochar is becoming. So maybe the interaction will be different. Of course, for use it as a horticulture substrate, you maybe want more active biochar. So it's really depending on, you have to look at the whole story. So maybe now it seems that producing biochars at higher temperatures, it's more promising for this application. But then, yeah, it might also have if you have biochar production at higher temperatures, it will also result in higher pHs initially. So maybe then you will need more sulfur to acidify. So yeah, I think it's a combination of feedstock and production conditions that you have to optimize. That's what I call the fine tuning.
And I guess you will also have to have a fine tuning for different substrates for different applications. So making it
Yeah, because now we started with a quite difficult crop, let's say, because it's this azalea, it really prefers a very low salinity, but also a low pH in the substrate. So it was like a worst case to see what happens there. But maybe with strawberries or other crops, it can really work very well already with the material we have now. Yeah.
Another question was from Christian Freirichs. Did you use water or gypsum extract to measure the EC?
It's a water extract. Yeah.
Okay, so if there are any other questions, please write them in the chat. You have another minute or so. Rian was just mentioning that they burn the vinegar at TNO to because of the phytotoxicity of vinegar. I guess that's more of a European way of dealing with the vinegar. It's not that famous in Europe. If Bart is still around, if there are any questions coming up, feel free to answer. Otherwise, I say thank you very much, Bart. Great presentation.